Bismillahirrahmanirrahim. Assalamu alaikum. My name is Maryam Akhtar and I'm from the Department of BS Chemistry. Today I'm here to represent the topic of my practical, which is proximate and ultimate analysis of coal. So, before moving toward my main practical, I would like to tell you the theory of the analysis of the coal, which I will cover proximate analysis and ultimate analysis. In proximate analysis, we're gonna determine moisture, volatile matter, ash yield, and fixed carbon pattern which are present in the coal. Whereas in the ultimate analysis, we are going to determine the amount of the major elements that are present in the coal. For example, carbon, hydrogen, nitrogen, sulfur, etc. So let me move toward in-depth of the proximate analysis. The most common coal analysis is called proximate analysis. In first, ash yield, the material in the core that will not combust. Fixed carbon, the remaining carbon in the coal after combustion. Moisture, the moisture content that are present in the coal. Volatile matter, the compound that will be converted to the gas in coal during the combustion. These are the determination that we are going to discuss in this practical. This is an analyzer, proximate analyzer, in which the sample cups are placed in the analyzer for the automatic measurement of the ash yield, fixed carbon, and moisture and volatile matter. In case of ultimate analysis, it provides a convenient method for reporting the major elemental content in the coal. So, total carbon. Total carbon in the ultimate analysis is the measured weight percent in the carbon present in the coal. We use ultimate analyzer for the calculation of the total carbon which are present in the coal. So to determine the inorganic carbon and the organic carbon components of the total carbon sample, a split of the sample should be analyzed in the colorimeter, and then the carbon measured in the colorimeter is the total organic carbon, which is represented as TOC. With the colorimeter, measure of the total organic compound and the ultimate analysis of the carbon in the coal. These are the formula. Total carbon is equal to total organic carbon plus total organic carbon, which are present from the colorimeter. These two are the further calculation through which we can determine the total carbon in the coal. Total hydrogen. It is also measured by the weight percentage of the hydrogen in the coal. The hydrogen gas is liberated from the coal during the combustion as the water vapor, which comes from the organic compound in a coal, and thus to the correct measurement as determined, hydrogen from the analyzer to fuel-based hydrogen value is as follows. As hydrogen, which are fuel-based, is equal to the hydrogen as analyzer measurement minus 0.119 into moisture. This is how we determine the total hydrogen from the ultimate analyzer. If we are moving towards the total oxygen, this is the formula through which we can find the total oxygen which are present in the coal through the analyzer. Total oxygen weight percentage is equal to 100 minus total carbon weight percentage plus total hydrogen weight percentage plus total nitrogen weight percentage. Means all the compound elements that are present in the coal for the analysis of the other substance. Total nitrogen that are present in the coal. Nitrogen is found in the organic fraction of the coal. Upon combustion, it is liberated as nitrogen oxides in the fuel gas emission. Oxides of the nitrogen emission from the coal combustion are regulated in US. So, if you're heading toward the total sulfur in the ultimate analysis is measured weight percentage of the sulfur in the coal. Sulfur in coal occur in three forms: organic, inorganic, elemental. Inorganic sulfur occur mostly in minerals such as pyrite. Total sulfur is an important criteria in steam coal because sulfur combines with oxygen in boiler to form a sulfur dioxide. Now I'm talking about the latest method for the analysis of the coal. So the aim is to determine the sulfur and ash content in sample of the coal or a charcoal. And the chemicals are charcoal, barium chloride, hydrogen chloric acid, sodium carbonate, magnesium carbonate, and magnesium. And the apparatus are standard volumetric flask, etc. Theory. The residue left after the burning of the coal is known as ash. So the first step is ash content determination. Weigh the 1 gram of the coal in the weight in previously weighed silica crucible. The crucible is heated in a muffle furnace at 800 degrees Celsius for 200 hours. Disturb while heating. Crucible is weighed and ash content is thus determined. Now, determination of the sulfur content. It is the same as the ash content determination, but we have to add and mix 1 gram of the sodium carbonate and 2 grams of the magnesium oxide in the crucible. The crucible is heated at temperature and for 2 hours and cool the crucible and add 2 normal of the HCl. And after that, pipe it out 10 milliliter of the solution and add 10 milliliter of the 10% barium chloride solution to the beaker. Wash the precipitate with the cold distilled water until the filtrate is free from the chloride ions. Cool it and keep it in the desiccator for 15 minutes and weigh the precipitate as barium sulfate. The result will give out as percentage of ash in inorganic residue and percentage of sulfur, which is the total sulfur percentage in the coal. Now, this is the determination of the form of the sulfur in the coal. Total surface monosulfide, pyritic sulfur, sulfate sulfur, concentration that are determined in the whole coal samples. So what are the sample requirements? The whole coal samples are received as an air-dried 80 mash powder. Typically 500 milligram of the sample is required for the measurement of the total sulfur and the 4 forms of the sulfur. So this is done through the flame atomic absorption spectroscopy, 2 laboratory hot plate magnetic stir combination, and spot lattice frame to be used as a rack for the equipment for the form of the bottom flask. So these are the matter for the total surface content present in the coal. Total surface content of the sample is determined by use of the sulfur analyzer. Calibration of the sulfur analyzer is based on one of the eco coal standard. After calibration, we do the procedure. And the released sulfur dioxide gas passes through the infrared absorption detector of the sulfur analyzer. After integrating the absorption signal over a period, the instrument explores and prints out the concentration of the sulfur. Laboratory setup for the separation of the forms of the sulfur. 10 3-necks round bottom flasks are placed in the heating mantles. The first and the fifth flask has thermometer in the side of the neck. Nitrogen bubbles through each of the solution and the bottom of the flask and then passes through the condenser and the test tube. This is the monosulfide sulfur separation and determination of the sulfur content. First, we have to place sample into the round bottom flask. Then we have to add distilled water into the sample. And after that, we have to add zinc acetate into the beaker. And we have to place a magnetic stirrer in the solution. And after that, NaOH is added, but do not boil the resulting mixture. And then we will add the mixture of the zinc acetate into the test tube and bubble of the nitrogen through the system for 15 minutes to remove the oxygen. And switch on the electrical power to heating mantles. Vacuum filter each alkaline zinc acetate mixture through filter. Discard the filtrate. Place the filter on a marked watch glass. Place vanadium pentaoxide over each precipitate to determine the concentration of the sulfur in a bag. So this is how we determine the monosulfide. Now this is the turn of the sulfate sulfur separation and determination. Vacuum filter the solution in which the round bottom flask through the filter. We have to wash the residue by using HCl, and after that the residue in the Allen-Meyer flask and save the determination of the pyritic sulfur. Place the filtrate, add distilled water, And then place the beaker on the hot plate and bring the solution to the boil. Add barium chloride to the boiling solution and set the beaker on the large hard plate and evaporate the solution to the dryness. Means we have to place it for overnight. Place the filter paper and precipitate on the watch glass. Subsequently, we have to add the vanadium pentoxide over the precipitate, and this is how we will determine through the sulfur analyzer. The last one is the pyritic sulfur separation and determination. We have to add distilled water to the residue of the Allenmere flask, and after that nitric acid to each flask, and then watch glass on each flask and set the flask on steam bath. Vacuum filter the mixture in the flask and wash the residue. And this is how I determine the iron leach from the sample of the nitric. And then calculate the pyrite sulfur, which are as follows. This is how we will calculate the percentage pyrite sulfur in ppm. This is the calculation of the organically bounded sulfur. Percentage organically bounded sulfur is equal to percent total surface sulfur. So that was all from my side. Thank you.